Good morning. Thank you for coming. So this will be my usual functional variation talk with some new results as well. And what I'll try to do, I will try to root the problem of the functional annotation of genetic variants into needs of human genetics. So we'll think about function from the point of view of human genetics. And I always start my presentations with referring to debate between biometricians and Mendelists about 100 years ago, and you probably know all about that from your Genetics 101. So Mendelists considered genetics to be simple with traits like in case of the P here, or maybe human disease segregating in pedigrees, right? So we have individuals expressing phenotype, individuals non-expressing phenotype or expressing different phenotype, and there are segregations according to Mendels Law. Now, this picture was seen to be contradicting to experience in evolution, experience in agriculture, experience in the analysis of general populations. So biometricians considered this an oddity and postulated that real genetics looks like this. So you look at the population, maybe I don't know, students at this campus, or fish in the pond, or corn in the field, and you see genetic variation, which results in phenotypic variation, frequently quantitative traits, or maybe binary traits that do not segregate according to Mendels' laws, but they still follow certain statistical principles, which I'm going to talk about in a second. So this contradiction has been resolved, of course, again, as I will be explaining in a couple seconds. But what is important for us is that thinking about functional effect of variance, we think about two different types, about alleles that would segregate in populations and alleles that give rise to this traits which we now call polygenic. So we'll think about there's two functional classes. It's important to think about them, because genetic problems are different, so interpretation of genetic results are different in these two contexts. And on the other hand, functional effects of the variants may differ as well between these two scenarios. I'll start with simple Mendelian case. I don't know how many of you attended MPG presentation by our group. We discussed Mendelian genetics applications, and the usual situation nowadays is that we have a family with, for example, here unaffected parents and an affected child, and we only consider scenarios of either dominant or recessive, fully penetrant alleles, right? So if there is presence of the variant, there is presence of the phenotype. So if this allele would be dominant and fully penetrant, and parents would be unaffected, the only possibility is that this is de novo mutation. We can now sequence 3 genomes, and if we focus on protein coding regions, and this is a big if, and we'll talk about that, however, most people do, so we presume that alleles of large effect that give rise to this particular phenotype are coding alleles, then on average we would see a single event in the child, right? So every human would have maybe 60, 70 de novo mutations, de novo point mutations. If you restrict yourself to protein coding regions, that's roughly 1, and the distribution is textbook Poisson distribution. So if it's roughly 1 in average, you may have 0, you may have 1, 2, maybe 3. You're very unlikely to see 4. So this means that you generate very few variants, coding variants, and you have to analyze them from functional perspective to see if this is a likely causal variant or not. Now, another possibility that these variants are recessive. In this case, one would be inherited from father, one would be frequently different one would be inherited from the mother. Now, these numbers are not rooted in population genetics as on the previous slides. They are more rooted in the way we analyze the data, because usually we only consider very rare variants. Again, our assumption is that these variants have very large effects on the phenotype. They are deleterious. They may not reach high frequencies in the population. So depending on filtering stringencies, we would take maybe 3 to 5 compound heterozygote single nucleotide variants Maybe 1 or 2 homozygote variants if parents are unrelated. Sometimes you would have a structural variant. Again, it's a very small number of variants, very manageable, but you have to select the right variant. In this case, when there is an affected mother and affected daughter, we would roughly see 50 variants shared on average, right? So this is, we would think it's a dominant transmission. And for a dominant transmission, again, this depends on stringency of our filters. We would see roughly 100 very rare non-synonymous variants per individual. Shared between mother and daughter in this case would be 50. Our experience is no biology can get you out of 50 variants, right? So usually a good biologist could connect any gene to any phenotype, would give you a very plausible story. And we tested it multiple times. You cannot get out of 50 variants. However, in these cases, in dominant cases, you may have extended families. And if there is, for example, an affected cousin, then there is a possibility to bring the number of variants down to, say, around 10, which is, again, somewhat more manageable number. So in this pedigree, we would sequence 2 cousins. We would not sequence these individuals because they are obligate carriers, so they would share every single allele these 2 individuals share. If pedigree is more extended, we can probably interpret the variants on purely statistical grounds. Usually this is not the case. You don't have enough individuals. So you would bring this down to, say, 10, 12, maybe 8 variants. Again, that you have to sift through. Having this number of variants, you have to analyze them. And usual principles are that you look at known genetics, human or animal, and see if these findings can be supported or contradict known genetics. You look at the gene function, both reading literature and with automated methods, looking at things like gene expression, maybe interactions, and so forth, and again trying to resolve whether this function supports the role of the gene in this phenotype or contradicts it. But then there is a question which I'm going to focus on today. What is the chance? So imagine that the gene makes sense. What is the chance that this particular mutation would disrupt function of the gene or alter the function of the gene? Would be loss of function or gain of function? How do we know that the variant is in the gene makes sense, maybe it does nothing to gene's function, right? So this is the most important question. How can we tell whether this variant has a functional effect? And of course, you have to always think, what is the chance that my observations happen by pure chance? So this is the problem in modern Mendelian genetics, where you have a few variants. You believe that your causal variant is among the small set. You'd like to guess which variant is causal and get some evidence in support of that. And here is where you turn to function, to annotation. Now let's talk about complex traits or quantitative traits, right? So why is the problem different? Well, in case of, for example, human height, what we know that if we line up many individuals, we observe the distribution which resembles normal standard Gaussian distribution, right? This is statistical property. This property is surprisingly common for a lot of human quantitative traits. You would look at blood pressure, bone mineral density, cholesterol levels, and so forth. You would observe roughly the same distribution. Another argument is given by this plot, which was originally provided by Galton, is now called Galton plot. What's on this plot is height of parents versus height of children. It's mid-parent height. And what you see is that joint distribution of parental height and height of children is approximately bivariate normal. So if the distribution is bivariate normal, we don't have just variance and mean, we also have covariance, and we have this regression line, and in case of bivariate normal, regression line is linear. And we see that children of taller parents are usually tall, and children of shorter parents on average are relatively short. So we can think that maybe this trait is due to genetics, right? We also know that children of tall parents are usually not as tall as their parents, and children of short parents are usually not as short as their parents. And this is what Galton called regression to mediocrity. And again, this shows that height is probably only partially genetic. And we can compute how much of height distribution is due to genetics. So there are multiple statistical properties. There are more that were analyzed by biometricians, and again, they thought that quantitative genetics, or genetics of binary complex traits, or there are also countable traits, is a different beast from standard Mendelian genetics. Now, this debate was resolved, as many of you may know, by Fisher in this paper in 1918. So what I usually tell at least students in my class is that there is difference between studying sciences and humanities. If you're studying humanities, you're told to read original text by Aristotle or somebody else, and you go through the text and you try to analyze the text, right? If you study sciences, you're given a textbook which has very well prepared for you modern understanding of state of science, which has very well interpreted modern language results, which are classic scientific results, right? So I think this paper is an exception. It is worth to read this paper, like you're in humanities class, the original text. A, because it's a great paper. B, because our model of quantitative trait variation or complex trait variation is still the same as Fisher came up with that in 1918, right? So we have tons of technology, tons of sequencing instruments, not as many new ideas. So the basic model is that if you think about quantitative trait, you would think that many different loci influence the trait, right? Some may increase value on the trait on average, others decrease. And every locus is segregating in Mendelian fashion. Now, for us now, it's trivial that discrete alleles, single nucleotide variants, insertions, deletions, or other changes in DNA are discrete events, and they would segregate in the Mendelian fashion. How Fisher got this idea, I don't know. Somehow he guessed it right? So every locus is segregating in the Mendelian fashion, and the phenotype can be modeled as, for example, additive over locus effects plus non-genetic contribution. We frequently call it environmental, and this model immediately gives rise to normal distribution, to Galton plot, to many observations of biometricians, right? Many statistical observations on trait variation. So this is a great model. Now the question is whether effects of those alleles are similar to the effects of alleles we were talking about looking at Mendelian cases, right? And also how many of the variants. Is it the same problem? Is it one variant, two variants, three? So similar to the problem in Mendelian genetics, or this is something very different? Some results point to models of extreme polygenicity. Very many very weak effects. Some postdocs in my lab with background in physics call it infrared catastrophe, right? Infinite number of very small effects. So what are reasons for us to believe that these alleles have very small functional effects? So the first types of experiments pointing in this direction come from experiments on artificial selection. So there is persistence of selection in populations. Now, I always tell the story, even though it's very hard for me to believe in it. This is plot from review by, I believe, Peter Kittling. It's same numbers in the text and numbers in the plot. In the original paper is the same story. Again, hard to believe. So this is artificial selection experiment in Drosophila, because it's much easier to do selection in Drosophila than in cattle, right? To do experiment in artificial selection. So the setting of the experiment is the following. The selection is for speed of flight. So Drosophila, well, I usually ask what Drosophila eat, right, in group studying genetics. So Drosophila eat yeast. Yeast lives on fruit. So you release flies, and they fly towards banana, right? And you measure how quickly they fly, and speed was 2 centimeters per second. So if you imagine Drosophila flying, that's about right. Then you take flies that reach banana first, breed them, and then put a fan blowing wind in the opposite direction. So now these flies have to battle the wind, right? Some of them can do it. You take these flies, breed them again, ratchet up the fan, increase speed of wind, again see how many flies can still reach the banana, take them, breed them, go to the next generation, right? So you go through 100 generations, you go up and up and up, so you start with flies flying at speeds of 2 centimeters per second, you end up with flies flying over 180, so almost 2 meters per second. Okay? Again, it's very hard for me to believe in the existence of these flies, I cannot imagine them. I don't understand why Department of Defense doesn't fund Drosophila research. But this is what we're observing, right? Why is this telling us about allelic architecture of the traits? Because imagine that there would be only 3, 4 loci that are responsible for speed of flight. In this case, we would see rapid saturation, right? You fix one locus, another locus, third locus, and you would not get any additional response to artificial selection. The model that may easily explain persistence of the response is the model which is highly polygenic. There are many loci of weak effects. Or another possibility, you pick up new mutations on the go, but this would always also point to a very large mutational target, right? Many genes, not very big individual effects. Another example from relatively recent PLOS genetics paper, this is about chickens, right? Many generations of artificial selection, many generations. You start with a chicken weighing 1 kilogram, 50 generations of selection, you get a chicken which is twice as heavy, right, 2 kilograms, and you get another one which is half kilo. Okay, in 50 generations, you change weight of the chicken twofold, in one direction or the other direction. And then you can do genome-wide association studies between these two lines and see what are the effect sizes contributing. And most of them, I think the largest is 150 grams. Most of them are very weak, very small effects, right? So from mathematical approaches and artificial selection and cattle breeding, some of the statistical methods were brought to human genetics, for example, by Peter Vischer's group, Michael Goddard, Young, and others. And these methods also support highly polygenic architecture with plenty of weak effects. The idea here is that you fit a model which is infinitesimal, so a lot of effects, very weak. Effect sizes are drawn from normal distribution, and they explain fairly large proportion of heritability in the data. I'm not going into statistical details, but again, this is some of the methods to analyze polygenic traits, and they again point to many small effects, very many variants of very small effects. Now, what are potential functional roles of these effects? We thought that for Mendelian traits, people look in coding variation, people sequence exomes. You think that this variant should disrupt protein coding parts of genes. Here, it is possible, and I'll talk more about that at the end of the talk, that most of functional variation is non-coding. And when I'm saying non-coding, non-coding is not a positive statement, right? It means that it's not in protein coding part of the gene. Mostly we have no idea what it does. We think that it is involved in gene regulation, so this variance can be in regulatory elements such as promoters, enhancers, silencers, insulators, all of those elements. Right, so they change the way how genes are regulated. Okay, so now the impression we get from data is that traits that are Mendelian and traits that are complex polygenic are caused by different classes of variants acting through different biology. Are they caused by different genes? We don't know for all the traits. However, in many examples, there is very good overlap between Mendelian phenotypes, which being extreme versions of complex polygenic traits. I think the best example we have is from analysis of blood lipids, and this is from review by Seika-Therezen. So if you would look at loci that cause Mendelian syndromes, for example, hypercholesterolemia or Tanger disease, so lipid disorders that segregate in families in Mendelian fashion, and you would look at findings from genome-wide association studies, you would see that there is a lot of overlap, right? So almost every single gene involved in a Mendelian syndrome produces a signal in GWAS association study, right? These signals also map very well into known biochemical pathways. So for example, in high-density lipoproteins, so HDL cholesterol, all the genes that push cholesterol from cells, package, enzymatically alter, absorb in liver, all of those genes produce genome-wide association signals, and also many of these genes, severe mutations in them produce Mendelian disorders, right? So at the level of pathways, these signals are frequently the same. Again, I'm not saying always. I give you examples. The biology is always more complicated. But we have a number of examples where Mendelian syndromes involve the same pathways as related polygenic traits. However, functional effects of variants are, differ. Okay, so now I switch gears and I move to actual functional part of the presentation. And I'll start with coding regions and severe effects, and then move to this regulatory weak effects. So first, what do we have? What tools do we have in our possession to prove causal effect of a variant on the trait, right? Well, statistically, we can look at segregation of the variant with the phenotype, right? So see if certain allele co-segregates with the trait. That's very good. There are, however, a couple problems. One is the allele gain should be a very strong effect to see significant segregation. Second, you need very large pedigrees, and very frequently large pedigrees are not available, at least in, I'm talking human genetics. Another problem is sometimes the inference is wrong because something can sit in the same haplotype and linked regions is pretty big, so you don't know whether what causes segregation is this variant that you're analyzing and not variant on the same haplotype. In vitro functional data, again, very attractive. The problem is that for many proteins, functional assays don't exist, so you have to develop an assay. Also, when you're measuring function in vitro, you don't know, you measure some component of the function, right? So if you measure, for example, enzymatic activity, but the problem is with cellular localization, you may not detect it. So you don't know whether you're measuring function relevant to the phenotype in question. Also, you don't know quantitatively how well functional change is translating into phenotypic change. Is that the case that very small change in your functional assay would translate to dramatic effect on phenotype, or very large effect would translate into small change in phenotype? You usually don't have this information. So these are functional assays. Again, very valuable. I'm not saying they are not valuable, but every method has problems. Animal models is in vivo models is a great resource, very laborious. Again, you have to establish phenotypic assay in a model organism. This assay should be specific, should resemble human condition, right? So I tried to randomly match sequencing data in humans with zebrafish phenotypes, and when you're talking about things like, I don't know, autism or anthropometric trait like waist to hip ratio, is very difficult to model in the fish, right? So then you can look at reference frequency in controls, and of course, in case of partial penetrance or small effect mutations, that may not be very informative. This may be informative for highly penetrant variants that are strongly deleterious, or for phenotypes with very low prevalence. The problem here is that presence in controls at high frequency usually rules out the hypothesis about causal relationship between the variant and phenotype. The contrary frequently is not correct. So if you don't see the variant in number of controls, you don't know whether it's functional. Now, the human population with the current size is mutation saturation experiment. When we sequence enough people, this would be enough, but we're very far from that point, right? And then there are computational methods that I'm going to talk about, so bioinformatics. It's cheap, easy to run. Current methods are not tremendously accurate for reasons, again, I'll talk about, but the problem is so severe that we're still in business developing them. Okay, what are types of the variance? So first, when we look at the variance, we look at specific annotations. So if we'd like to say that the variant is protein coding, it means that there is a transcript I believe is expressed in my cell type of interest and so forth. Sometimes in the annotation you'll know there are multiple transcripts, there are conflicting annotation. One annotation will tell you that the variant is synonymous, another one that it's a missense, the third that it's non-coding. Be very careful. That's very important. So be careful with conflicting genomic annotations. When you look at the variants, usually the class of variants people are mostly enthusiastic about because it's most likely to be of functional significance are nonsense variants, or also frame shifts. So variants, protein truncating variants, so variants that are presumably loss of function. And this is great. For this class, mostly you can rely that the variants alter function of the gene. However, there are certain caveats. First, they are enriched in sequencing artifacts. Not because there are more sequencing errors in nonsense variants, but simply because nonsense variants are very rare and sequencing artifacts are uniformly distributed. So the proportion of sequencing artifacts among nonsense variants is a little higher. Then the considerations to pay attention to are what's location along the gene. Does this variant cause nonsense-mediated decay, right? Not all variants do, so you should look where the variant is located, and whether the variant is in a commonly skipped exon. So if there are alternatively spliced isoforms, this exon cannot be present in most of the isoforms, and this nonsense variants may alter only a very small fraction of transcripts. But generally, having a nonsense variant in your genetic study as a discovery is usually considered to be good news. Another class of variants are variants involved in splicing. So of course, variants in canonic donor acceptor sites usually disrupt splicing. However, there are variants that affect, more subtle variants that affect splicing, and they don't reside in canonic sites. Those would be in exonic or intronic splicing enhancers. Those are much more difficult to deal with. There are computational methods. We rely on Spliceman by Welfare Brother Group at Brown. There are other methods to predict the effect of variants on splicing. There are variants that create splice sites. Those are very difficult to predict and guess because they would be somewhere in the middle of the intron. There would be almost perfect splice site, and mutation would make it perfect. And then there is a splice isoform which knocks down the function of the gene. So Spliceman, the idea is to work on sequence entropy. So the method basically depends on sequence entropy analysis. But then there are many other considerations. So the splice change shouldn't alter the frame, should alter the frame, is more likely to be functional if it doesn't lead just to exon skipping, and so forth. Right? So there are additional considerations rather than strength of the splicing site. Splice mutations, there are mini-gene assays where you don't need patient material. You can perform them in the lab and experimentally test the effect on splicing. Okay, the largest class of mutations are in protein coding regions are missense changes. So those changes that change amino acid. They substitute one amino acid for another. That's very difficult because some of them are completely benign, some of them are equivalent in the effect to complete loss of function, some of them are hypomorph, so they are of intermediate effect sizes. So that's a hard problem. So many groups, including ours, have been developing methods to predict the effect of these changes using evolutionary information and protein structure information. What is the basic idea? So I'll start from evolutionary side of things. Imagine that you have your human protein, And then you have protein sequences from related species, for example, orthologous sequences. And I'm not going to discuss like whether you have to include paralogs or not. So let's think about a simple model where you have a bunch of orthologous sequences. Then you look at your site of interest, and you're wondering whether variation in this site is consistent with the mutation you observe, right? So I make two assumptions, right? One assumption is that this variation, so what is bad for human, is also would be bad for dog and fish and other species, right? So in evolutionary terms, this is the assumption of constant fitness landscape, right? So I assume what's bad for fish is bad for human. I also make an assumption that evolutionary signal is informative for what's happening within the population, right, or within individuals. So if I make these two assumptions, it's still a complicated statistical problem because the data are not independent. They are connected by phylogenetic tree, which has to be taken into account. Also, if there are multiple amino acids, for example, well, we want to know ideally for each of 20 amino acids what would be the possibility for this amino acid to be inserted in the site. And we don't have enough species still, so we have to use Bayesian methods of Bayesian statistics to develop the methods. Okay. So if we make these assumptions, then we can estimate whether mutation fits the pattern of amino acid changes, and we can estimate the rate of evolutionary change at the position. Again, I assume constant fitness landscape. What's good for me is good for fish. What's bad for fish is bad for me. And there are multiple variants. This may not be the case, right? So one is environmental. I cannot live in water, and fish doesn't give primer tox ridiculously early in the morning, right? So we live in different environments. Another reason is that fish has different genome, so there are many other changes in addition to this mutation in question that may interact with the effect of my mutation. Okay, so if I make this assumption, then the model is very simple. So I start with some codon encoding glycine. It's called continuous time Markov model. I wait for a certain amount of time. There is a change. May not change amino acid, but then it may change amino acid. And I can feed this model to phylogenetic data and try to analyze whether frequencies of different substitutions and the position seem that they would allow my mutation to happen, and I can analyze the rate. So in little more rigorous statement would be that what we're interested in estimating stationary distribution for this Markov process and the rate with which it happens. Another source of information is, of course, protein structure. And here, great observation is that most pathogenic mutations affect stability rather than specific functional sites, right? Why is this happening? This is happening because mutational target is much larger, right? So there are many more ways for me to change stability of the protein than to alter specific binding or some specific change. That seems like a good news because you can look at protein stability.
Can you explain what you mean by stability?
Yeah, so what I mean by stability. So in this case, I mean delta delta G. So there is change in free energy between folded and unfolded states. It's called delta G. Delta delta G is change in free energy due to mutation, right? So I introduce mutation. The effect of mutation can be that I energetically stabilize or destabilize folded state, or I can stabilize unfolded state, for example. So I can push the equilibrium towards unfolded state from folded state. So I don't generate stable structure. So this is the parameter of interest here. The problem is, so it looks like structure provides so much information in addition to sequence. The problem, unfortunately, is that it's very difficult to estimate. We tried pretty hard. I know other groups tried. You can always build predictor of delta delta G that would correlate with disease status of your mutation, for example, but make it a useful predictor is very difficult. Instead, at least in our method, we rely on heuristic structural parameters and use them together with evolutionary signal, and they can boost evolutionary signal, right? So you get additional information from structure, but primarily from heuristics, not from careful biophysical considerations. Again, if there is somebody who thinks he or she can solve this problem, we will have postdoctoral opening in the lab for this type of activity. Our method is polythene 2, so we take sequence-based information, structure-based information, throw into probabilistic classifiers, currently naive Bayes. We had version with support vector machine and so forth, and return predictions. There are many other methods available. Very small subset are listed in this table. Again, easy to run. Bioinformatics methods always easy to run. The accuracy, well, it really depends who measures accuracy and how do you measure accuracy and what's your training data set. I would say around 80%, 75, 80%. Much better than random, a lot better than random. Not really useful, say, in clinical genetic diagnostics, so you cannot really rely on this as a sole source of evidence. Why is it difficult to use structure and evolutionary signal to build very good prediction methods? There are two reasons in my mind. I'll talk about evolutionary reasoning. One is this assumption whether differences between species are informative about what's happening within populations. And the problem here is that many variants within populations we believe are weakly deleterious or moderately deleterious, right? It's very difficult for such a variant to be fixed between species. So again, very standard population genetics, if I would look at selection coefficient, so selection coefficient of say 10 minus 4 means that on average in some population due to this mutation for individuals without mutation would have 9,999 progeny versus 10,000 progeny for individuals without the mutation. So the chance of the mutation with this effect or larger effect to fix between species is pretty much zero, very small, right? So it means that comparative genomics has a problem with resolution. I don't know whether my mutation is lethal or causes Mendelian disease or reduces fitness by 10th of a per mil, right? So by 10 minus 4 or even 10 minus 3 wouldn't be of interest to any practical, say, medical problem. On the other hand, if it's 10 minus 6, it behaves totally like neutral, right? So it can fix without any problems. So, well, this is assuming effective population size of humans. So the problem is here that there are weakly deleterious mutations that can segregate in the human population without big visible effects, but would never fix between species. You would predict them as very deleterious mutations from comparative genomics. Second problem is this assumption about constant fitness landscape. So what I'm assuming is that this mutation in this genetic background has the same effect as in this genetic background and on some completely different genetic background, right, if I'm using comparative genomics. The reality may be that in some backgrounds mutation is deleterious, but if I go to a very different genetic background, it's completely benign. This is, again, back about constant fitness landscape. This problem is related, if you're into speciation theory, to Dobzhansky-Müller model of speciation. There are many interesting consequences of dependency on genetic background, which we usually call in genetics epistasis. So how this can be seen in practice. In this paper, published I guess over 10 years ago now, with Fedor and Alex Kondratrovs, we studied many examples like that. This example is hemoglobin. Look at human hemoglobin, and at least the database we never checked, but the database says that substituting value number 20 for glutamic acid produces disease phenotype, right? And then you look at multiple sequence alignment, and you see that horse, and this is the reason I had horse in my previous slide, has exact glutamic acid here, right? So there is structure of horse hemoglobin, and what we think may happen is this glutamic acid interacts with this histidine 69, which is likely to be protonated, nicely formed salt bridge, right? You look at the human structure, instead of histidine, here is glycine. Glutamic acid probably wouldn't fit. Again, this is speculation. I'll show you some experimental data where we did check things. But there is speculation based on structure that the effect of this glutamic acid depends on the interacting residue in 3D. So recently, in collaboration with by Nika Katsanis and Daniel Jordan, a PhD student who recently left the lab with Stefan Frangakis at Zug, so they did the study, we looked at all available mammalian genomes and even all available vertebrate genomes. And what we're seeing is this. So if I take a variant which is annotated in human as neutral human polymorphism, for overwhelming majority of these variants, I see some other vertebrate carrying this exact variant in this position. What happens with human disease mutations? So this is what is interesting. There are many technical issues I'm not going through because quality of disease databases is not ideal. But if we take databases that are not observed in the human population, are observed in multiple databases, seem to have evidence of high penetrance, even within mammals, we have 5–6% of this variants observed in at least one species, right? So the hypothesis we had is this is mostly because of genetic background and frequently because of changes in the same protein molecule. Now the question is, when I'm saying genetic background, is this collective effect of multiple substitutions along the genome or along this protein sequence that collectively compensate for the effect of pathogenic variant? So maybe, for example, they overstabilize the structure and one mutation which reduces stability is allowed, or they change the energy landscape and the structure, right? Another possibility is a single 1–2 events that specifically suppress the effect of the mutation. So we thought about the following model. So imagine that this is time, evolutionary time. So I go back in evolutionary time, start from human. And first question is, OK, I take variant of interest, say it's neutral. I go and back in evolutionary time and wait until this variant appears in somebody else, among us mammals or among us vertebrates. And what would be distribution of this waiting times? And of course, we think the distribution would be exponential. It's Poisson process. We just wait. And these are data, and this is the fit. And at least for standards of computational biology, this is pretty good. Very good fit. Now I take disease mutations, do the same experiment, and it doesn't fit. Right? So if I would take human mutations annotated as disease mutations, would do the same experiment, would go back in time, would analyze distribution of waiting times, that's not the case. Then you can come up with a different model. You can say that, OK, there are a number of changes you have to wait for, so they would collectively compensate for the effect of your change. Right? So I can write simple statistical model like this and estimate how many other variants I should see before my disease mutation is allowed. So this model fits the data quite OK. But what was mostly surprising is the target size for suppressors is pretty small. And in most cases, one other mutation is required to completely suppress phenotypic effect. Right? It's a little hard to imagine. Again, this is the model. So most people wouldn't buy it. So a person who took it seriously was Nika Katsanis at Zug, so we decided to do in vivo experiments. So what we could do, we could take the phylogeny, we could predict the specific set of compensatory changes, would be a large set. But again, in Nico's lab, they can inject a lot of different changes. And the experiment we proposed is this. So for example, they measure this phenotype, which they believe resembles fish phenotype for mutations in human syndrome called BBS, Biddle-Bartlett syndrome. This is the fish embryo, this is the egg, and they measure this angle, okay? And this is wild-type fish. They knock down the gene, and they see change in the phenotype. Then they take, it's important, human mRNA, inject human mRNA in the egg, and observe rescue, right? So this angle is reduced. And then they look at human gene with the disease mutant, and if mutant is functionally significant mutant, this mutation would not rescue, right? Its effect would be identical to knockdown effect. What we suggested, we take a double mutant, and if there is no change between single mutant and double mutant, there is no suppression. If there is a rescue with double mutant and no rescue with single mutant, this is a suppressor. So we'll look at the data, and this is one of the BBS gene, BBS4, and this is the specific change. So you look at negative control. So it's qualitative analysis, so they have normal fish, class 1 and class 2. This is percentage of normal fish is in blue. So if you take uninjected, most fish are fine. You knock down the gene, most fish are not fine anymore. You inject human mRNA, there is good rescue, not complete, but pretty good rescue. And then you inject disease mutant, again, very similar to positive control to knock down. And then you inject double mutant, no effect. In most of them, except there is this change that offers complete rescue, right? It completely suppresses the effect of first mutation. It's a single change observed in other species, right? Another example where you have two partial rescues and again one complete rescue offered by double mutant. Again, one change completely suppresses the effect of mutation. Well, these are known disease mutations. Can you do it in real time? So going forward, this is a patient, unaffected parents, affected child here with a bunch of phenotypes. Microcephaly is among them, developmental delay. So we don't know whether it's dominant or recessive, so both models are tested. All genes were, well, except Titin. Titin is always a false positive for compound hets. It's the longest gene in the human genome. Other genes were tested in fish. ETG2 is a gene with very relevant biological function. And the phenotype in fish recapitulates human phenotypes. So you have microcephaly. Neuronal expression was tested. And again, the gene affected neuronal expression. And the quantitative effect, quantitative assay on cell proliferation also showed the difference. So the interesting part is this mutation reverts to mammalian ancestral background. It's valine to methionine. Valine is primate specific, right? Outside of primates, everybody has methionine, including zebrafish. Same story. We take all mutations on the mammalian ancestral background. Two of them completely rescue the effect of the original mutation in the fish phenotype, right? So what we think happens in human case is that this variant in primates, this mutation would be pathogenic. It would not be pathogenic outside of primates. This makes predicting based on comparative genomics very difficult. Again, as I said, we don't know enough structure. Comparative genomics, you have to develop multi-locus models. Again, we don't know how to do it yet. This is on the prediction. So I think I have 5 more minutes to go through non-coding variants. So these are all stories about coding variants of large effect, what we're dealing with primarily in Mendelian genetics, but also some of them in complex trait genetics, especially in sequencing studies, in rare variant studies. All these methods are relevant. What about non-coding variants? So again, variants in all of these regulatory elements, proximal and distal, and we think that they are involved in regulation of transcription. There is a tremendous progress offered by epigenomic projects like ENCODE, projects like Epigenome Roadmap, measuring epigenetic marks, histone modification, DNA methylation, and DNA accessibility. So these marks can guide us towards the location of regulatory elements in the genome, right? Those elements that are responsible for transcription. Why do we think variants in these regions are functional? Well, one type of signal comes from, again, the analysis of natural selection. There are many regions that are conserved in phylogeny, and if you look within human population, whether there are signals of selection, natural selection within the human population, in these regions you detect the signals. This is statistical observation. Genome-wide association study signals are primarily located in non-coding regions, and they are enriched in putative regulatory features as evidenced from these epigenomic studies. So these are three pieces of evidence suggesting existence of many effects, mostly likely small effects of variants in these non-coding regions. Now, saying that, the story is not clear yet. We have no idea about biology underlying conservation. Conservation doesn't overlap. I mean, there is enrichment always, right? But it's not like regulatory regions would always be conserved, and GWAS signals would always be located in regulatory regions or in conserved regions, right? There is a famous experiment by Nadava Hituf and Rubin and Pinocchio and others when they did genetic experiment on ultraconserved region, deleted it from a mouse, and mouse is perfectly okay. So there is no 100% overlap between evidence from evolution, evidence from genetics, evidence from function. What we do know, however, is that this is a figure from Adam Sipel's paper. If you look at regions of open chromatin and binding sites for transcription factors, and look at the affinity of sites, the inference of natural selection does correlate with information content of these motifs and with affinity of binding. Right, so there is some correlation between evolutionary signal and functional signal. We know that GWAS peaks are enriched in certain regions of regulatory regions highlighted by epigenomic projects. For example, here, if we sort all SNPs by p-value in genome-wide association study for QRS, how many people know what QRS is? Some do. I'm not a doctor, I'm just playing one on CV. So this is phenotype related to EKG, to measure of electric activity of heart. And what we see here, there are more of SNPs with p-values in the low range in regulatory elements, putative regulatory elements, specifically in the heart tissue, right? Not in other tissues. So there is enrichment which is cell type specific. This paper by Shomurai Chaudhuri, he developed a method where he locally, with Gosha Trinka and others, moved location of the peak around and measured enrichment in genome-wide association studies for different phenotypes versus cell types and versus different epigenetic marks. And again, what they see is that if you look through the phenotypes, you look at LDL, the enrichment is specific to liver. In rheumatoid arthritis, the enrichment is specific to immune cells, and so forth. In type 2 diabetes, for example, both pancreatic islet and liver produce these peaks. And yeah, this is exactly what my plan was. So slightly more sophisticated analysis with partitioning heritability from Lebofalkis Price, Sasha Gusev and others. What they show is that heritability explained by functional regions is strongest in this putative regulatory regions enriched in open, marred by open chromatin. So again, if you look at coding regions, the enrichment is 13-fold with respect to size of the region. Coding regions are important also in quantitative traits, right? However, the total, because they are small, total amount of heritability is explained by regulatory regions is much greater, even though the enrichment is only 5-fold, right? But total amount is much bigger. So one of the directions the community, 2 slides and I'm done, the community is thinking about is to change this paradigm from GWAS peak to controlled model organism system and to biochemistry to a method which I call human genetics all the way. When you take GWAS peaks, look at endophenotypes, various endophenotypes, which many of them would be molecular, for example, gene expression, and then you can do methylation, chromatin accessibility, and so forth, and come up with this effect on molecular phenotype. So with this consecutive changes on phenotypic definition to more and more and more down to molecular level, this approach may offer possible functional interpretation of GWAS peaks. So I'm done. Just always show my lab and how seriously we think about our projects. There's some original work in this talk, and this was done by some member of the lab and also great collaborators. Thank you.